Welcome back. I hope all had a great summer. As you know, this is Bioinformatics 8060. This is our speaker seminar course in bioinformatics. And so today we're starting off our fall 2026 semester with Dr. Ethan Pickering. So Dr. Pickering recently joined UGA in 2025. He is an associate professor in the Department of Crop and Soil Sciences, as well as in the Institute of Crop Design, where he leads the Computational Agriculture Lab. In this lab, he does research on building AI and computational methods for better understanding and improving biological systems, specifically for crop design. And so in addition to being an associate professor here, he also has recently founded an AI for agriculture startup called Profit AI. And before coming to UGA, Dr. Pickering actually was at Bayer Crop Sciences, where he led AI and data science research. His most recent title being the head of Global Research Data Science Hub. So he has a very technical background, as you will continue to hear. And so before he joined Bayer, before he started his tenure at Bayer, he completed his postdoctoral fellowship at MIT in mechanical engineering, where he used AI to model or to predict rare and extreme events in complex physical systems. And before that, he sort of stayed in this domain of mechanical engineering. So he completed his bachelor's from Case Western Reserve University in mechanical engineering, and then followed that up with a master's and a PhD also in mechanical engineering from MIT, from Caltech, where he specialized in computational fluid dynamics. And something interesting about Dr. Pickering, which I just found out about yesterday, is that he was actually raised on a family-owned vegetable farm in Ohio. So despite his very technical background and focus on engineering and mathematics, he's sort of done a full circle in life and come back to his agricultural roots, kind of is like fulfilling his destiny in a way, it feels like. So it's nice to see that. And yeah, he's very passionate about using his technical background and employing mathematical approaches to, like I said, try to model complex biological systems and try to solve some challenges that are faced by modern agriculture. So he's sort of had an unconventional path to come into the crop and soil sciences, but I think those are often the most interesting ones and sometimes offer the most fresh perspectives when you're trying to solve a problem and you just have your domain experts. Sometimes you need that outside perspective. So I'm very excited to hear his talk today, and I hope that you all are as well. Before I begin, before I hand it off to him, I should also say that this is also kind of his kind of a part of his application to become a courtesy faculty in the Institute of Bioinformatics. So if you're a part of IOB, definitely give him your undivided attention. Even if you're not a part of IOB, definitely give him your undivided attention. I'm sure he's going to talk about if he's interested in taking graduate students, if he's interested in collaborations. So if you fall into those categories, definitely give some good feedback at the end and ask some good questions. So with that, everybody please join me in giving him a warm welcome. And he goes on.
Wow, it's really impressive you memorized all of that. I sent it like last minute yesterday, so I thought for sure this would be a read thing. So yeah, thanks everybody for coming. I'll talk a little bit about that background as well here. I had a title, I think it was a slightly different title, but today what I'd like to talk about is how we can think about agricultural AI and how we need it to bridge various different scales between genotype, phenotype, and environment, in particular in what I like to call the sparse data limit. I'll give a little bit of some definitions of what those mean, but also a lot of this work was I've been here at UGA for about 7 months, so a lot of this work is work that I was doing back at Bayer Crop Science. So, oh, that was too fast. So what are these different definitions that I'm kind of referring to in relatively a broad way? So we've got genotype, which is going to be the genetic makeup of an organism. So we're looking all the way down to its DNA, and I like to think about this at the nanometer resolution. And then we have phenotype, and these are a little bit, some air quotes around this a little bit. So these are broad definitions, but some measured attribute or trait of an organism. And this is going to be around your meter level in many cases when we're taking phenotypes. And when we think about environment, these are all the different external conditions that affect an organism, and they tend to be interesting along, or at least for crops, we care about them at the kilometer scale. because if a crop doesn't perform at the kilometer scale, then it's not really a crop. And then also in the sparse data limit, what does that mean? That means that we have the number of samples that we have to train these models are going to be drastically smaller than the input dimensionality. And that's a really difficult modeling task. We'll talk about that in a little bit. So as was mentioned, a little bit of kind of the background here. So I started on a family farm in agriculture, went through this route of engineering for a bit of time. It's taken me. So I was doing mostly things in mechanical and aerospace engineering throughout my bachelor's, PhD, and postdoc. And so I'm really more of an engineer. So I'm going to say a ton of things that are incorrect, most likely from a biological perspective. I just don't know better quite yet. So still working on some of those concepts. Then went off to Bayer before coming and kind of seeing this as being some trajectory here of from an engineering perspective, a dynamical system, which I think a plant and a crop is a dynamical system that we need to model. So what are the big, what's the big problem that we're looking to solve here? Well, agriculture must adapt faster than ever. We have a growing population of 10 billion by 2050, and that's going to require an increase of nearly 60% in agriculture. And so and there's challenges all over the place in terms of climate and changing growing conditions, disease spread, regulations that we have to care about, and doing this in some manner that's going to be sustainable for the planet, as agriculture is about 20% of emissions today. So some components of this is if we think about drought tolerance and looking around and seeing that 14% of global crops experience drought in 2022. If we look at questions around disease resistance, there's a corn disease tar spot that emerged in about 2015, and over the next 10 years has ended up going all the way across the entire US. Creates tremendous damage. We also have soil versatility issues. So when we're designing crops, where the soils that they have existed are now going to need to shift up based on the days that are required to grow them. And then we also have other aspects of extreme events that are interacting with these crops and creating massive losses. So here's an example of a flattened field out in Iowa. This is a 2020 derecho that did about $2 billion of damage across corn and soybean. So if we were to stay at the same levels of capability, this is going to require that we have arable land the size of 2 Indias to meet that 60% increase. And so that's just simply not going to be the answer. And so we're going to have to think much differently about how we design crops. So now agriculture, what is really difficult though is that agriculture is global, it's very diverse. So the environments that agriculture grows in and the way that individuals grow their crops is very diverse. And so that means that we can't have just a one size fits all solution. But there's an opportunity here, and that data can uncover some of this variability, and we can use AI to bring some of that diversity as opportunity to action. So one area that I like to think about in terms of where you see agricultural data increasing in scale, it's increasing in scale, resolution, source, and type, and this is going to demand likewise changes in how we think about modeling these systems. So I think on the genetic side in particular, how you sequence for markers over the last really 75 years, we kind of started at pedigree information. We've seen base pairs in the early 2000s, and this is economically viable at scale. So something that you can sequence at scale and make use of it in an agricultural pipeline. And we're getting to this point where we're less than $1,000 to get a complete high resolution DNA sequence for something like corn. And so I think this is a really massive opportunity for all that information that we're uncovering, but we need to move also on the modeling side to actually bring all of this into value. So we also see this on the transcriptomic side. We see it in gene expression data. We see it in gene ontology. And there's a similar story when we think about the soil data we have, the weather data that's available, management data, phenotyping, particularly imaging, and I think is very, well, I guess lunch is happening soon, and present at IOB. All right, so I guess we get reminders. Let's see if I can kill that or not. I don't know. Here we go. dismiss. Okay. So now, because all of this data is all in different types of structure and sources and types, really we don't really have so many models that really understand how to deal with all these different different types and such. And so that's where we really have to turn to AI modeling and the different opportunities that presents us for learning these very weird structures, data sources, and putting that all together. And so when we think about how agriculture, how we have to design this sparse limit, we need to design a particular genotype that's going to perform in an environment to achieve a very specific phenotype. And so on the input side, if we think about the total genotype that we might have, we're on the order of 10 to the 9. So we have billions of base pairs that can potentially affect our outcomes in phenotype. When we think about environment is technically an infinite dimensional system because it is these functions that are forcing and interacting with our organism that we care about. And when we look at phenotype, we end up seeing that we might only be collecting one variable. We will be collecting it at only a handful of samples. So some of the biggest data sets that would exist at a place like Bayer for yield is going to be 10 to the 5th. That is really not nearly enough data to truly build a model that goes in between that can map genotype and environment down to a final phenotype. So I like to argue that the data scale here is out of balance. So you have all this G data, you have all this E data, and you have very little phenotypic information to build between these models. When you think of a lot of the foundational AI models that are out there, they're really very balanced in terms of their inputs and their outputs, and that's what makes them be particularly interesting and be particularly useful. And I would generally argue that for the agricultural perspective, and maybe even on the plant biology side, that data scale will always be out of balance because we have particular issues with the infinite dimensionality of environment and the incredible variability that exists on the genetic side of things. It's effectively infinite as well, even though technically it's not, but it's effectively. So we need to think differently in how we design these models. So what I'm going to talk about today here is a number of different concepts of how we can use AI across these different scales to start help us rebalancing this data scale. And so there's 3 different scales that I've mentioned. We've got phenotype at the nanometer level, phenotype at the meter level, environment at the kilometer level. And we can do, on the AI side of things, we can build new architectures that can work well in this data sparse limit. We can build loss functions that work well in the data sparse limit. And we can think about how we interpret our data differently if we know we don't have so much plentiful data at our disposal. Couple of things that we'll get to in this. So we've got on the genotype side, that's what we will start. We have an architecture we've been developing called AgRem, which uses regulatory elements in it, and also this idea of cascade, which allows us to look across single cell data to find motifs. And then a fun one here in the middle that is one of my favorites is bioinformed neural nets, or we call them bins. And then finally, some large-scale simulations that really come more from the Bayer side, but I think there's opportunities to build those in the public domain as well. So we'll start here with genomic scale, and a lot of this is all going to be based around concepts of large language models and loss functions, data interpretation, and some architecture. So jumping into these cell type specific losses. So this has been some work that we've been doing in collaboration with Bob Schmidt's lab, where they have some really great data sets. In particular, we've been working a lot with a soybean single cell atlas, and Ali Fargadan, who's a postdoc in my lab, has been working on this topic. So we've been using some backbones from foundation models and starting to build different concepts and architectures around those backbones. So we haven't really been interested in building a new backbone, but building other concepts around it, so that way we can match it up with some of the data. And some of the things that we wanted to make sure that we could include were things around different tissue types, different cell types, utilizing as much ACR data as we can. One thing that we started to realize as we built this architecture and we started to look at some of the data was that we ended up finding that it was pretty biased towards a number of housekeeping genes. So we wanted to ask the question, could we build some loss functions that really cared about some of the more rare genes that are not always on in every single cell type? So we built this model, and kind of off the bat, we just want to know how would its general predictive capability look like. We built and trained this model, and here's actually two different types of tissues. This is the Pearson correlation coefficient, and this is the cell type. We ranked it based on different Pearson correlation coefficients. A couple tissues are, this tissue is more difficult than other tissue there. And then this is more or less what we're looking to do is predict, and here's our observed data and our predicted data. We're getting mean across gene PCCs of about 0.68, and then area under the curve, we were getting very high values. This is typically what's reported as very high, but it depends how you define your metrics. Models look better if you use one metric. If you use another metric, it doesn't look so good. Soybean in particular has been a very difficult one for us to get individual point-wise by point-wise RNA expression at the single cell level. That is a very quantitative metric and a very difficult thing to do, but it's incredibly important if we want to actually design to very explicit gene expression levels when we are designing a new crop or a new plant. So when we look across these those different cell types, and if we look at kind of the bulk aspect, so here's us pushing all of that expression together. So this is what it looks like in the bulk side of thing as we then expand it out towards different cell types. There's this question of how can we differentiate against these different cell types? And there are certain genes that are on all the time. These are your housekeeping genes. And there's other genes that are only on a very specific few cell types. So we started to ask the question, how do we define this particular distinction? And so what we started to say, or really this is kind of a way to describe this as being very cell type specific, just a couple different cell types a gene is on in. This one is moderately expressed, and then we have something that is a housekeeping gene. So this is some level of activity where expression is beyond some amount of threshold. And so if we look at the histogram of all the different genes and what we are defining as the breadth, so if you are on all the time, here's all your housekeeping genes. They are in every single cell type. And then as we go down here, these are your very cell type specific genes that are only on in a couple of those cell types. And so as we looked at our mean squared error of this prediction, as you go from the very cell type specific down to the housekeeping genes, for a couple exceptions that exist here, generally your error is getting much, much better. And the reason why is because there's so much more data that is biasing you towards creating solutions and creating better predictions for your housekeeping genes. We might be more interested in a number of these cell type specific. So it performs worst for the cell type specific, far less data from the model to learn from. So how can we adjust this? So we took two different approaches. We call one the frequency-based weights and then activity-based weights that we use to essentially rescale that bias. So that way we can learn more about the cell type specific genes that we're showing our model. So this is a little bit more difficult to look at. So this is mean squared error, our breadth index, and then we have our different MSE losses as we change from a mean squared error loss to activity-based losses and frequency-based losses. This is really that MSE improvement that exists. So all here, those two different types of loss functions improve the predictive accuracy for your cell type specific. But then what's really, really key about this idea is that there is no free lunch. You will make it worse in your housekeeping side. So you have the same amount of information, but you just get to tell your model what should you pay attention to and what should you not pay attention to. That's the general idea there. So we are okay in losing this information such that we can get that information. And what's also key about some of this is that you can learn things. You need to keep this data around because there is some useful housekeeping information that is important for also being able to predict cell type specificity. So you don't just want to throw that data away. You need to find a way to deal with it appropriately. So a couple of cool things that happen when we change this this loss function in the data. So here in the top, this is for the MSC loss, we have a learned embedding. And so what is this kind of TSNE plot we're showing? So, or a UMAP plot. We're showing this is the ground truth gene expression across these different cell types. And you end up seeing these kind of cell type specific islands that exist. And then you have a number of and this is also colored by breadth. So these are very cell type specific. And then here's your housekeeping up here. When you create a model based on your MSC loss, it makes this really continuous map. All models are trying to do this. They don't really like to they like to spread things out. They like to keep things continuous. They're not super happy with all of these islands. And so you get a learned embedding and you have a predicted expression that is relatively continuous. Oh, not that far. And as we use this activity-based loss, you start to create these cell type specific islands. So you're starting to force the model to respect some of these concepts that are disjointed in some latent representation. And so this is kind of a fun thing that we can look into the latent space and see how these loss functions start adjusting the learning of these models, and in particular the latent features that you get after you push the sequence through a foundation model. So a couple things we can then do with this. So here we did some in silico mutagenesis of these different base pairs, and we were able to start looking at what does the in silico mutagenesis change when you go from an MSC loss model into a different loss. And so we end up seeing that there's particular regions and certain cell types that end up having a very one-sided or a very marked change in how the in silico perturbations adjust. And so that's one sign that we can look at that loss function for changing the results, and we look at that change, and we can say that has something to do with cell type specificity. So there's still some more work we need to do on this. These are still some preliminary results, but that's just a little bit on this concept of some loss functions. Now we'll move to this concept that we've been thinking about called cascade, which is context-aware significance of cross-gene attribution for discovering elements. We just put a bioRxiv preprint out there, and this moves from from our loss function side now to some of the data concepts here. So I was mentioning this in silico mutagenesis, where we substitute every base pair with three different alternatives. So depending on what base pair was there, you give the other alternatives, and you're looking at the sensitivity and how that is adjusting predictions for your expression. And so using that same model before, we had about 40,000 genes across these 66 cell types. And we want to ask more carefully, how do we understand some of the seclets or motifs that are existing in these different structures? And so as we were doing some of the loss function, we started to realize that maybe there was also an issue in the way that people were interpreting seclets or motifs across these different concepts of cell type and some of the locations that we look at from the transcription start site. And one of the things that we noticed that the most common way to find these different seclets or motifs is what's called TF-MODISCO. And so TF-MODISCO looks across all the attribution and creates basically just a threshold. There's this threshold that exists, and if your attribution scores go above this, then you are a candidate seclet that you can then look at that seclet and then see how often that appears. And if it appears a lot, then we say that's a very interesting motif. But if you look at where positionally from the transcription start site, where most of the TF-MODISCO motifs were coming from, it was all upstream. And we were much more interested in understanding some of the promoter differences that exist upstream. And this would be downstream here. We were really interested in understanding what seclets are coming from here and what motifs exist here for the purpose of editing questions and such. So we found this was massively biased. You would get really the same motifs over and over again. So we came up with this idea of cascade that creates a variable threshold that exists that's conditioned based on position. So that way it's not necessarily looking only at this one very high region of sensitivity, which for many reasons is sensitive. And so we've also noticed this in other pieces of literature where Some models have used TF-MODISCO on one of their, this is actually the same lab that created TF-MODISCO. They also created a paper with Chrome-V-Net, and they found that their motifs were massively biased. And so then they did a ton of different things to try to fix that. So they created architecture shifts to make this happen. And so we were doing a different data analysis technique to make these shifts happen. So this TN5 motif is strong and sits essentially in every single peak. And so that was popping up there. When we do this, we're able to start shifting. On the left side, this is TF-MODISCO, and then this is Cascade. We were able to start shifting all of the discovery of these different seclets and or motifs to the left side and upstream in the promoter. Now, these results end up being pretty similar to some degree, about 0.8 correlation. So it's not a very different method, but it is shifting the way that you're making your interpretation and where those exist along the gene of interest. And so this kind of shows just very, maybe more clearly where Cascade is end up finding, this is your median seclet position relative to TSS. And so if you have MODISCO, they're all here. We're creating something where we're allowing it to really explore a little bit further away, particularly upstream. And when we do this, we're not only doing it with position, but we're also asking the same exact question across cell type differences. So we can go and ask, okay, are there particular motifs that seem to be gene specific? So they are existing across every single cell type in one gene. They could be global, so they exist in every single gene. That's also a possibility as well. And then tissue specific is that they're only existing in one tissue, and then cell type specific is there's one particular cell and a particular tissue that motif is particularly interesting. And we can kind of count all of these up. And there's a lot of different mining that we need to do to understand some of the implications of these different seclets. So here's an example of us running this and showing a cell type specific sequence. So there is your seclet that pops up, another tissue specific. So this happens across all of the cell types in that tissue. And then for a gene specific sequence, which is across all of the different, across all cell types and tissues in that particular gene. So that's a little bit about how we've been starting to interpret some of the data from these large language models. And the next part here is how do we start changing the architecture so we can add in a lot more information. So we've been particularly interested in how do we add in some regulatory elements into these models, because right now they were just sitting plus minus 2,000 base pairs from a transcription start site. So we created what we call AGREM, this attention-gated regulatory element module. And really the really important piece here, I'm not even going to go into all the details, is this particular the AGREM module that allows us to take distal elements, kind of chop them up in a computationally efficient way, and then push those through to enhance some of our expression predictions at a single cell level. In doing this, so we've applied it to both some human cell lines and then also soybean, because we were really looking for nice benchmarks that exist. And on the human side, we found more benchmarks. So we were comparing this to a couple different models, Espresso, EP Informer, with a number of different components, and then our AGREM concept. And these are for two different human cell lines. And then we also apply it to soybean. We think there's a lot more to go in soybean, so we're not exactly super happy with our AGREM results quite yet on this side, but we tried a number of different types of models. But what's important about the concept of AGREM is that you can use many different types of element sets that you can get into the very similar architecture here, which allows us to chop these up and basically feed all of that sequence information from distal elements far away from the gene. All right, so that was everything on the large language model side. That's our nanometer resolution side of scales, and we were trying to get that to expression. Now we're going to talk about some markers, expression data, and then field phenotypes that we can apply this to. We'll start off with a loss function concept here, and these are really more breeding models. And so this loss function is around rare alleles or rare markers. It's really more rare markers. So if we look at our typical predicted and observed graph that we would use to understand the different errors of our model. Where here, this is the predicted perfect prediction line. This is over predicted. This is under predicted. Typically, we would use a standard mean squared error loss function to basically push all of our data to that perfect prediction line. But when we look at a lot of the data that will exist in the field, we end up having this distribution and this histogram where what we really actually care about if we're designing a crop is we want it to either be at any of these extremes. So we want to have more yield. We want to have more disease resistance. That's the real design goal. And so you have a loss function that actually doesn't care about the design goal, just cares about getting the mean correct. So this can sometimes lead to two different behaviors. This is the more rare behavior, but it's very bad, is where you have very poor tail prediction, but you have very good prediction in the mean of the distribution. But what's more common, what you get in many models is you get compression. So you end up that your tails end up being compressed so much that the predictions cannot span the distance of the observations. So you don't understand what your tails are at the end of the day. So we wanted to add in this, create really a targeted tail prediction where we are okay with understanding these tails, just like we were kind of doing in the other approach. And we will give up on the mean, but we want it on both sides of the tails because this will tell us what alleles and what markers are good and which alleles and markers are bad. So we use some of the ideas from some of the postdoc days to use these, what we call likelihood weights, to balance the learning in the sparsely populated tails. And you can show via some theory on how this works and how much it weights your model to understand the tails and how much it gives up in the median or in the mean. All right, so here's an example of some data that we had at Bayer here with North American tar spot. So tar spot has this 20 to 60 bushel per acre effect when you get tar spot in areas of corn. And there's a couple, I don't know if this was supposed to have an animation, but it's not there anymore. So this is how tar spot spread. So tar spot started around in north central Illinois in 2015. And then over the next 10 years, rapidly spread as it looks like a lesion or a pathogen that's just spreading out radially. And there's some big issues with this. And as I was mentioning before, we have different growing bands that exist across the US. So as you move from the south to the north, you have very different population structure that exists in corn. And an issue with that is that if you have all this different population structure, you have alleles that aren't going to be accounted for across typical models. And as we look at some of the data that we were capturing here, 9 is really susceptible and 1 is the resistant lines that you want. And so as we were building models, this tail problem was a real issue. So we don't care about that, we just want to make sure that we can understand this heavy tail. Yeah, I was mentioning that there's a million different gene versions that are difficult for, say, a 10,000 biallelic SNP genotype. It really can't understand all those different rare alleles that might exist that really aren't accounted for. So if we take our standard GBLUP, genomic best linear unbiased predictor with a mean squared error loss, you end up seeing that the predicted tar spot only predicts 5, 6, 7s, and 8s. And our observed tar spot is all around. This is a confusion matrix that kind of shows that, so you can see that compression in the model. And as we apply this other loss function, we're able to extend out that prediction, so that way we are able to now span the entire set of observations. So we now get some 1s, 2s, 3s, 4s, and we can more correctly find some of the genes and alleles that are important for disease resistance. So we get 3 times more genotypes that are discovered with these rare event losses. And GBLUP, basic GBLUP. Now you can use GBLUP with this likelihood weighted loss. It's perfectly fine to use that with a linear model as well. But particularly this MSE loss rejects diversity. So there's this opportunity to help out with some of the bias. All right, so now moving on to the architecture side of things, and this is probably one of my favorite topics, which is bioinformed neural networks. So if you look at what most models are used for breeding in agriculture, you're going to be using the Genomic Best Linear Unbiased Predictor. It's a great model for a number of reasons in terms of the different data that was available at the time, and still to this day is a very good model and very difficult to beat in many cases. But you're really looking at a situation where you have a very underfit model. You have very limited information, and as you build just a genetic linear model with also some fixed environmental effects, you're really going to underfit most of the data you have available to yourself. On the other hand, if you just use a deep learning model that's off the shelf, you're generally going to get lots of overfitting because there's just way too many parameters that are there, and you're not going to be able to generalize very easily. So is there some way that we can find a balance between those two things? This was the very first thing that when I went to Bayer, that was one of the challenges, was how could we use some of the domain knowledge that exists? So things like explicit gene interactions that we know about. Could we use gene regulatory graphs? Could we use gene expression? Could we use other aspects that we understand about biology that we have very high fidelity information, but we might have very little data about it? So we only have a couple RNA-seq experiments, we have a ton of markers. And so on this other hand, you have data at scale. So you've got maybe 10 million phenotypes of yield, and you have over 100,000 unique genotypes that are in the marker setting, but it's very coarse information. So you have this coarse marker information, and you're also just missing other components that are important for the biology of a plant. So we wanted to then ask, could we combine that and call this a bioinformed neural network? Now, there's a couple of reasons why this makes sense to do in a couple different ways. So first, we mentioned that off-the-shelf AI models are over-parameterized, but they're also going to struggle with long-range interactions. So if you know two particular genes are going to interact with each other, they're 100 or they're a billion base pairs away, no matter what model you have, no matter what attention you give it, it's going to struggle to learn that concept. If you already know it, get it in there. So we can increase the sparsity, we can reduce complexity, we can call out these known interactions, and there's also an opportunity that it starts to become interpretable because we're trying to actually emulate what biology looks like compared to just put it into a black box. So we just had this, I don't know why it keeps doing this crazy skipping. So we just had a paper, our bioinform model just became published in the Plant Journal, and we were working, and it was a massive effort by Katyana Kontalati, who's a senior ML researcher at Bayer and was a member of the team. And we were working on two different questions. So how could we use a maze TWAS data set and build a bin around it? And then also we tried a synthetic shoot branching problem, really just to show some of the theory around bins. So I'll show a little bit of both. So the concept of bins is that we start with some omics selection. We create some phenotype linkage between some of those omics data. We create sparsity mass, which is really this idea of how many of these connections can we throw away from the model and which ones do we need to keep. We can build that type of model architecture that has this amount of sparsity in it, and then we can use that for either genomic selection or also potentially for trait discovery, as we can perturb the different layers that exist in a bioinformed neural net. So a quick example of what this looks like on the TWAS data. Okay, yeah. So here we use marker data as our first layer, and then we used RNA-seq data to derive what we believe to be this gene intermediate layer that existed, and then also which genes are then connected down to a final phenotype. And in between these layers are what we call baby neural nets. They're just these little teeny tiny neural nets that the only thing that they're responsible for is learning very basic elemental relationships instead of having everything fully connected. So there is this massive sparsity that exists between the markers all the way down to the phenotype. Now, after we build this bin with the RNA data, we actually don't train with it. So we're only using the RNA data to help us build the architecture, and that's it. Then we don't throw it away, but we put it to the side. And then we actually train the weights of the model by giving it just the marker input and the final phenotypes. So we build the architecture with some domain information, and then we actually train it with just the G to P data and let it take whatever form it needs to take. The reason why we care about doing this is really what we're trying to understand is, what is the relationship? Is it a nonlinear or linear relationship between the G side of things, the bioinformed data that we have, and then also that final phenotype? So here's 4 different cases that can generally exist if you're doing GWAS and TWAS, or maybe you're using a bin. So case 1 is that you have linear. In this case, GWAS will work because you'll have linear relationships all the way through to the end. In the case, also TWAS will work because you have this linear relationship. In the case that you have some nonlinearity between your marker set and your, say, gene expression here, then GWAS won't work. That will break the whole chain. TWAS certainly will work, and can also work for that. And then the big issue that occurs is if you have a nonlinearity between your gene expression and your phenotype, or both nonlinearities. So generally what you want to do if you're building a model, people ask all the time, you have a nonlinear concept and you want to be able to interpret, you build a nonlinear model and someone says, well, can you interpret it for me? And that's really hard to do because we don't really interpret nonlinear things. But what you can do is if there was a nonlinear function and you were able to interpret it perfectly, and you could see that you're predicted and observed, or you're able to model it perfectly, and your predicted and observed were 1-to-1, then you would say, well, the interpreter exists in that model itself. So in that black box, that's where that exists. And so in the cases where we're able to perfectly learn both sides of this, you should see that your domain information will correlate 1-to-1 with your latent variables. So that's an important thing for thinking about these intermediate nonlinearities that can exist. And so when we applied this to that RNA-seq data that was out in the field, so there's about 700 genotypes and 700 RNA-seqs on a very diverse maze data set at different populations. We were able to find that some of the latent variables that we had from that gene layer and the observed gene expression that was never used to train the data, just to help build the architecture, would correlate at high values. And so here we have about 0.74 correlation, and then another 0.74, 0.71. So we'd see this, we weren't really exactly expecting it, but when we ended up seeing that we had this high correlation between the latent variables and the gene expression, that gave us confidence that we were learning those two G to expression and then expression to phenotype layers. And with those genes that have high correlations, we then went back, we opened up that layer, and then we did a perturbation analysis to try to find nonlinear gene candidates, but only for those that had high correlations at that intermediate layer. So it looks somewhat similar to a TWAS. Those that are mapped here or called out, those are some of the actual, those are also TWAS hits that you get. So there's some extra hits that you get outside of TWAS, because you should also recover anything that comes from a TWAS. All right, some other concepts around, this is now on the synthetic side. So we were really interested in the very beginning. We actually started with the synthetic problem. We wanted to prove that a bin could work. Then it took us a very long time to find experimental data that made sense for us to go do some things on. So this was actually, this predates all of this work by a pretty good amount of time, where we were looking at how SNPs or genes could then could inform some different pathways of, say, auxin, sucrose, and then cytokines and strigolactins, down to what the phenotype is here is the time to outbud growth. So when you chop off a leaf, how long does it take until that bud comes back out? And there's a number of signaling that goes on for that to happen. This was particularly for roses and flowers. So there's more or less some established set of ordinary differential equations that show the dynamics of how these different pathways interact with each other. And what we want to do is, instead of use those equations exactly, we wanted to take those equations and start building a bin. So the way that this generally operates is that if we looked at, say, cytokinin here, what needs to connect with cytokinin needs to be auxin and sucrose. Those are in the equation. So those two things are connected in the architecture. We're not going to tell it about the relationships, all these different coefficients. None of that we're going to tell it, but we're just going to say auxin and sucrose need to be given in the bin to cytokinin. And the same idea with, say, strigolactin, you have auxin, and then for your downstream phenotype, this downstream phenotype takes sucrose, strigolactin. So you're building this domain architecture. We then add in some linear additive SNPs. So that way we have our description of our genotype input vector. We took a biallelic vector approach. So it's either present or absent, or you get one of the two, 1 or -1. We then had these intermediate variables that were scalar anywhere from 0 to 2. And then we trained on a number of, and we can also include these intermediates in our loss function. I'll show something about that. So that's one nice thing about building an architecture like this, is that you can actually help in terms of anchoring some of those intermediates. And so we looked to train this on, to test this on about 20,000 samples. We chose an input dimensionality here for the marker values to be 1600. And then we wanted to train with training samples anywhere from 500 to 20,000 for this concept. And then we also added in multiplicative noise on the phenotype, as well as some of these intermediate phenotypes. So we don't want just perfect data, we need to add some noise onto the phenotypes that we have, as well as intermediate variables, because we should have some noise in those intermediates. So to show how this performs against your standard GBLUP, so here we have on the y-axis is your normalized test mean squared error. And then on the x-axis, you have your training data set size. So with the GBLUP, we're going to have that as being our 1 right there. And as you get more and more data, it gets better and better. Then we'll just use a basic off-the-shelf over-parameterized neural network. As you get more and more data, that error improves. And by the way, the fully connected neural net will be better because this is a nonlinear problem. Those equations that we're looking at are nonlinear. So a GBLUP cannot, this is the floor of where a GBLUP can get. This, if you keep on going and going to infinity, will get to 0. Then as we start using the bin concept, we can massively reduce the error, particularly in the limited training data set side of things. We can also use what we call a hard loss, which is we actually start anchoring this data with some of that intermediate variables, or intermediate data. And then we also use what we call a soft loss, where we anchor them, but only with correlation. So we don't actually need the latent variables to be perfect. And what's super important about these results is that this green region right here, this is the region where your number of samples are smaller than your input dimensionality. And that goes all the way back to our sparse data concept, that we will always have less samples than what we have in terms of our input dimensionality. As we go to infinity, all these things collapse and it doesn't matter what you choose. And that's the case of massively plentiful data. But we will always be here, and I would say in the agriculture setting at the least. Also, we don't need full partial intermediate data for training. So this was a case where we only used, for all the different, let's say we had 100 of our genotype to phenotype information, we would only need 10 of the metabolic information. So you can have partial data. You don't have to have perfect G intermediate and final phenotype. You can have G, some of the intermediate values, and then your phenotypic values. You could also have G and your intermediate and no final phenotype. So all of those situations work in building one of these types of bins. Right. And then I had mentioned before about opening bin up and looking at those pathways and this idea of correlations. So we opened up the latent variables, and auxin, cytokinins, and strigolactins, they didn't have much of a correlation at those latent variables, but sucrose did. And if you go back to the paper that we got this from, this paper was all about sugar availability being the dominant pathway for, essentially the dominant pathway for output growth. So the most important one is the only one where the latent variables actually seem to care about learning that relationship. There's still a little bit of nonlinearity left in there, but that's okay. If we had infinite data, infinite data and also relatively large baby neural nets. So if you make the baby neural nets bigger, it can learn a little bit more. This is a super quick thing here on some optimal data acquisition. This is all some Bayer stuff, and we're really using some of the concepts around how we can discover and forecast extreme events in terms of what is the very particular data you need to bring in to your model to train your model. So not all data is made equally. There's some data that's particularly useful and some data that's very not useful, and definitely was a waste of resources. So when we think about how we build or how we design crops, we've got about 23 more cycles to feed to 2050 to feed 10 billion. And in our typical iteration scheme, we start with training some G to P model. We then go and choose new G's to go out and test. And then we will go take those, we'll test those, we'll observe the P, and we'll continue on our genomic discovery pipeline. And so some ways that we can think about how you make these different selections are super important. And so we were testing out some ideas at Bayer of how we start using some of these Bayesian optimization or Bayesian inspired optimization ideas with how you exploit your model and you explore your model. And these are some of those rare likelihood losses. So this is your traditional approach. You're just exploiting. You're always selecting the best. That is, most of the breeding models take the best. You add in a little bit of some variance in there or diversity metrics. But if we could use some of these Bayesian optimization approaches, how are we able to best learn that the performance space of genetics? So we applied this to, it's a little bit difficult to understand what's going on here, but here are very top performers. And we were doing 10 years of selections. And we were counting how many of the top performers we could discover out of over 100,000 different genotypes. And what was actually really interesting about this is that when we made our selections round over round, we were picking the ones at this extreme and a lot of them down here too. We were actually trying to expand the tail depth. So knowing what's terrible is really useful to understanding what's great. You need to have both of those tails to understand that. And so we could find the same top performers with 30% less discovery. We also were asking the same question on the environmental side. You change your acquisition function a bit, and then you ask, okay, all of our different environmental testing locations, knock all of those out, and you can still get the same accuracy with that data. So a lot of that data was redundant. Now we had the opportunity to do a historical analysis compared to a forward analysis. So we can show that there's opportunities to do this. How you make that decision beforehand is the more tricky piece. And so we're trying to show some concepts around that. All right, so some very last things, and this will be a super quick section, is thinking how we get to this environmental scale and some large scale simulations. Mostly, how do we think about building models that go beyond kind of our typical linear G-blow idea where we have fixed effects and a design matrix that's really one hot encoded for the most part when you think about environments. And so everything looks like these dots. Can we take more of a multimodal approach where we understand the different concepts of both soil and weather in terms of space and time? So we need to have a space and time concept. That's super important for understanding the connectivity between all these different field plots that exist. So we built this just very basic representation of that multimodal model. And here's a quick little look at what. So there's a couple of our testing plots, and then we'll overlay some of the different environmental grids that exist in terms of soil and weather. And then we put that all together and start making some predictions. So over your typical G by E split, we got about 30% improvements, some leave year out 15% improvements. Leave environment out, very, very, very difficult to do. Novel soil and weather, 5%, and that's maybe a bit arguable. So what this really ends up leading to is that we were creating this really massive footprint that you could now take all of this data that exists, you put this all into your model on your gene E side, and then you can create these very large predictions across the entire US of how you believe that particular genotype is going to perform across different soil and weather conditions. So some quick hits here. You can look at soil versatility. So great performance across much of the Midwest, elite performance in Iowa up here. You can find some of those standability issues. So that's the derecho from 2020. And we can also look at some things around, say, drought. So here's an example of drought in 2023 and being able to reconstruct those types of observables. Now, one big problem is we don't have all of that data. But luckily, I think there's some really interesting concepts here in satellite. The satellite imagery is improving so much. There's some opportunities for us on the public side to grab a lot of this data and start connecting it. And so we've started some work on thinking about peanuts in Georgia, where we're starting to look at some of the satellite imagery that's publicly available, building some architectures to fill the various gaps. Architectures that can link satellite observations to ground observations, some efficient data acquisition, and so so those are a couple of the different ideas of how we can start filling in from genotype all the way to environment, that we're able to start balancing this data scale between architectures, loss functions, data. And really the goal here is that if we can start to do this in that direction, that's really what enables us to design, and really it's a crazy concept of thinking about designing, enable a design where you can understand a base pair change and observe it all the way from a satellite. It's a bit of a crazy thing to think about, but I think as satellites get better and a lot of our technologies get better and as models get better, it's a real possibility. And from the dynamic side of things, it reminds me of the butterfly effect, which is, does the flap of a butterfly's wings in Brazil set off a tornado in Texas? So that's, I think, at this nanometer resolution, a base pair set off a change somewhere else completely different in the world. So thank you. I'll take any questions. I know it might be just a little bit over time here, and tons of people were super helpful on all this work, and you should take a lot of credit. All right, thanks.
Any questions?
Well, on the plot where you compare MSE of beans and Gibla, I noticed that whatever the error bars showed were wider for beans. Do you know why that happens?
Yeah, so yes, so that's a really great question. So bins are more difficult to train. So so in terms of they have these little baby neural nets. And so as you're going to do gradient descent and back propagation, there are some times where they'll get stuck in different areas. And so that's something that we're continually working on, of how do you unstick them, because they can take some pretty large variation. And I think in particular, you might be talking about the hard MSC loss. We don't really, the hard loss, throw that one up. We don't like the hard loss. The intermediate loss is much better. But yeah, we still have some large changes there. So, or large variation, and a lot of it goes to the parts of the architecture that you're training. They can get stuck and they go into different solutions.
So simply, they are coefficient errors, not that you are propagating more uncertainty from functions.
Yeah, I would say they're more weight errors, but probably need to think a little bit more about the propagation issues.
I'm curious about the interpretability part. I see that in silicon mutagenesis to see where the positions are more important, but what about the layers inside the foundation model? Did you get to see maybe using some lens-based method to see where the decisions were being made? Maybe in a very early layer, the decisions were already quite accurate. Then maybe we can cut off the forward part, and that can be very energy saving, which is a lot of people's concern.
Yeah, the latent space is it's a difficult beast to interpret. So we've tried some different things of changing latent space sizes and such. Now we put after the latent space out of the GPN, we put on an expression head that then interprets that down to those final expression values. We haven't really figured out beyond creating those descriptions that I showed there with like a UMAP. I can't think of too many other ways to interpret it very well. So we haven't gone and asked those questions quite yet. It's been more so from if the model predicts well and we can do sensitivity analyses, that is probably the better place for us to think about interpretability. At least that's what our current opinion is.
Very cool array of things. I mean, for the bins and things specifically about that intermediate layer of the represented RNA-seq, have you looked at examples where there's like large divergence between what the RNA-seq says and what your model says? It seems it's like maybe it's the expression in a different tissue or a different cell type than is what being measured.
Oh, we've not looked into that level of complexity quite yet. Of potentially saying that there's certain ones. I think what maybe you're mentioning is, let's say there's one latent variable that's super uncorrelated, but then in a TWAS it says it's a hit. Have not looked at that. But that's an interesting idea.
Okay, actually, go ahead.
That was a great presentation. Thank you. My question is regarding the optimization of the loss function. So would you mind elaborating on the rare species that were used in the data set? To which time point do they specifically belong to? Or is it?
Yeah, so you're talking about the tar spot. The tar spot or the tar spots example where there are some alleles that generally these alleles are hidden. They're actually hidden. So you have two in a biallelic model, you have two options, which allele is being chosen, but maybe there's another one that's sitting there that might be from an exotic line. So a lot of times with, say, tar spot, if you're a breeding company, you'll look in some of the tropics and try to find some examples where there's some tar spot resistance. And then you got to get those alleles into the elite germplasm population. They will be rare by definition compared to the rest of the population. So that was part of the motivation in doing that. And it just, with the current SNP panel you have, it's this idea that you have two representations, one has a really high phenotype and the other one doesn't. You don't have the input representation. How should you weight those two observations? Which one should you take as truth and which one should you not? So there's this kind of hidden variable. That's really what it's doing, is it's finding hidden alleles that you can't interpret from the marker side, but you can interpret from the phenotype side. That might have made it more confusing.
It is kind of better now. But just to follow up on that, what the observations collected belong to a specific time? I mean, do they belong to the 19th century? Or?
No. So these observations were over the last couple of years, so they were all around the same time. An issue with tar spot is that when you get a new disease, you don't know how to inoculate for it. So you don't know how to actually create trials. So these are all opportunistic. So the data was just coming from wherever we could find it. So it's like, oh, this particular trial location all of a sudden had tar spot, we're going to go take readings. So that was an additional challenge, that we couldn't set up a perfect experiment because we didn't have inoculation techniques right off the bat.
Okay, I think that's all the time we have for today. So let's give Dr. Miller thoughts.
He's talking about it.
Actually, if it's winning.
Yeah, it's all the stuff that I talked a lot about it today. And it's time for the other stuff, yeah. To be able to share something. Yeah, we got things on the cell phone stuff. We haven't even looked at that.
Yeah.